Hello aspirants. This video focuses on the invertebrate evolution, right from Porifera up to Platyhelminthes. The idea is not to memorize phyla separately, but to track how complexity builds step by step. If you follow that evolutionary ladder, every feature begins to make sense. The master classification grid helps us understand how all animals are grouped based on 4 key features: level of organization, symmetry, germ layers, and the presence of a body cavity or coelom. We begin with multicellular animals. Some, like sponges, have only a cellular level of organization and are mostly asymmetrical, so they fall under Porifera. As we move forward, animals with tissues and organ systems show either radial or bilateral symmetry. Radially symmetrical animals, like those in Cnidaria and Ctenophora, are still acoelomates, meaning they lack a body cavity. Bilaterally symmetrical animals show more complexity. These are further divided based on their body cavity. Flatworms are acoelomates with no body cavity. Roundworms have a false body cavity and are called pseudocoelomates. Finally, more advanced groups like annelids, arthropods, mollusks, echinoderms, hemichordates, and chordates have a true coelom. This step-by-step classification shows how animal complexity increases from simple to more advanced forms. Porifera sits at the very base of this ladder. The organization is cellular level, meaning cells exist but are not arranged into tissues. Symmetry is mostly asymmetrical. They are acoelomate and their habitat is mostly marine. A simple memory anchor given here is SES. Sycon, Euspongia, Spongula. If you see cellular level plus asymmetry, your mind should directly go to sponges. The real working system of sponges is the water canal system. Water enters through ostia, moves into the spongocoel, and exits through the osculum. Along the way, food is captured, gas exchange happens, and waste is removed. The key cells here are choanocytes collar cells with flagella that drive water movement. Two details that students often miss. Digestion is strictly intracellular. Fertilization is internal and development is indirect. The first major evolutionary jump happens here. Tissues appear. Cells are no longer loose. They are organized into functional units. With that, symmetry also changes. Radial symmetry appears. These organisms are diploblastic, meaning they have 2 germ layers. Another important feature is the gastrovascular cavity, which has a single opening. So this stage is about 2 things: tissue formation and radial symmetry. Coelenterates are introduced as organisms that can switch body forms. They exist as polyp, sessile, and medusa, free-swimming forms. This alternation is called metagenesis. Their defining feature is cnidoblasts, stinging cells used for defense and capturing prey. If you see stinging cells or cnidocytes, it directly points to coelenterates. Ctenophores look similar to coelenterates, but they are different in key ways. They also have tissue-level organization, radial symmetry, and are diploblastic. But their unique feature is comb plates, rows of cilia used for movement. Another distinctive trait is bioluminescence. That glowing appearance is not just interesting. It is a direct identification feature. Now we will look into some easily confusable facts. Ctenophores and coelenterates may look similar, but their reproduction is different. Coelenterates can reproduce both asexually and sexually with internal fertilization. Ctenophores reproduce only sexually and fertilization is external. A subtle but important point. In both groups, sexes are not separate and development is indirect. If you mix up reproduction here, you lose easy marks. The next big evolutionary jump is toward organ-level organization. With this comes bilateral symmetry. Bilateral symmetry allows directional movement, and that leads to the development of a head region and specialized organs. These organisms are triploblastic, meaning 3 germ layers are present. Flatworms are introduced here, and their body is dorso-ventrally flattened. This is where real anatomical complexity begins. Platyhelminthes are built for parasitic life. They have hooks and suckers for attachment. They can absorb nutrients directly through their body surface. Some, like Planaria, show remarkable regeneration. Their excretory system uses flame cells. Reproduction involves internal fertilization and multiple. This final matrix is your revision shortcut. Porifera, cellular level, asymmetrical. Coelenterata and Ctenophora, tissue level, radial. Platyhelminthes, organ level, bilateral. All of them are acoelomate. That last line is the key. Pattern complexity increases, but coelom is still absent across all 4. A good way to retain it is to read it left to right as an evolutionary ladder. If you can recall this table under pressure, most classification questions become straightforward.